Thank you, Vanessa. So it is with great pleasure that I mean, it's the first session that we will open for Hispanic Heritage Month, and you saw already in the beautiful banner that BINA does, the 5 speakers that will be speaking throughout this month of September and the month of October. And so we invited speakers that several of us are great admirers of. They have done careers, some of them away from Latin America, which is where Hispanic Heritage Month is celebrated in this context. So they're Latin Americans that have done their career away and have either Latin American or Hispanic that have continued to contribute to the development of science in the region. And it is with great pleasure that the first speaker is Carlos. So I first met Carlos. They put me in touch with him about his great achievements. So I heard about him from somebody else and the great achievements that he has done in terms of the twin labs. And I'll tell you a little bit more about it in a second. And I had the pleasure of speaking with him, and I was just impressed. I was touched. It was a wonderful conversation that we published at some point in Focal Plane. And so he agreed to be the first speaker of this series. And so I'll tell you a little bit about him. So he did his bachelor's at Universidad Peruana Cayetano Heredia, that's in Peru. He then did his master's at Universidad Nacional Mayor de San Marcos. And then he did his PhD at the University of California, Berkeley. He has focused, so right now he's a professor of molecular and cell biology, physics and chemistry, and the Raymond and Beverly Sackler Chair of Biophysics at the University of California, Berkeley. He has a strong focus in biophysics, and so as part of his, one of the biggest contributions, I think, is he has a twin lab. So he has a lab that basically mirrors what he's doing in Berkeley in Peru. And so that has contributed a lot to development of human resources and of exchange of expertise and access to resources between both regions. And from what I hear also, he has in general been very supportive to many Latin Americans going back and forth. He and his team develop and apply novel methods of single molecule manipulation and detection, such as optical tweezers, magnetic tweezers, and single molecule fluorescence microscopy. So he has trained already several people who are now independent researchers, and who are following in his footsteps in terms of this same passion to develop science and really opening access for Latin Americans in many different ways. He's also a Howard Hughes Medical Institute investigator. He has over 300 publications. I was having a look recently. And so yeah, it's with great pleasure and with great excitement that we want to hear more about his work. And I wanted to say, in terms of the questions and in terms of the approach, so the different speakers will have, during this series, will have different approaches. Some of them will touch about how their career was as Latin Americans abroad or Hispanic abroad. So feel free to ask questions at the end. It can be both scientific, or it can be if you're interested in that aspect as well. And with this, I give the word to Carlos. We're looking forward to your talk.
Thank you, Mariana. It's a great pleasure to be here today and to present this talk. I'm going to be sharing my screen, so you allow me that. Okay. And can you see my screen?
Yes, you need to swap displays.
Oh, swap displays. Okay. Yes. Is that okay now?
Almost.
Almost.
Try the button again.
The button. Oh, I don't. Okay, here.
Yes. Maybe try exiting presentation mode.
Exiting.
And show and start again.
Stop share.
Stop the presentation mode in PowerPoint. I think it was not sharing to Zoom correctly.
Okay. Let's try again.
Yes.
Let's try again. As often is the case, machines play tricks on us.
Indeed.
Okay, I'm still trying to, okay. Let's see. Oh. Share. Okay, and then share.
That looks perfect. Thank you.
Great. Okay, sorry about that. Well, my name is Carlos Bustamante, and I am going to tell you today about work that we do in my laboratory to study molecular machines in the cell. And so I want to start by first mentioning that many processes in the cell require directional movements, and these directional movements are actually performed by tiny machine-like devices that operate as molecular motors. What do I mean by molecular motors? Well, molecular motors are machines that convert chemical energy in the form of maybe bond hydrolysis, for example, hydrolysis of ATP, or chemical gradients, for example, a gradient of protons, into force and or torque and displacement. So we can also define molecular motors in a more restricted manner, and we can say that molecular motors are enzymes that couple the catalysis of a downhill chemical reaction, for example, the hydrolysis of ATP, to the performance of an uphill task, for example, a mechanical task. In this way, molecular motors function as energy transducers, converting chemical free energy into mechanical work through the generation of force, torque, and displacement. Now, the cell has many, literally thousands of molecular motors, which are involved in a number of tasks. For example, during sporulation in Bacillus subtilis, the mother cell has to pump the DNA from its body to the pre-spore, and it does so at great speeds, 6,000 base pairs per second. This second motor that I'm showing here is the ATPase synthase, which is the molecule responsible for synthesizing ATP in our mitochondria, and which also can use the energy of ATP to generate a gradient of protons. And finally, I show here in the third movie, kinesin 5, which is involved in chromosomal segregation during the division of the cell. All these are examples of molecular motors, and they are using different forms of energy, chemical energy, to convert it into mechanical work. Now, how do we go about describing the operation of molecular motors? For that, it is convenient to define a space that I call the mechanochemical space. In the mechanochemical space, as shown here, you have on the z-axis the free energy. In one coordinate, let's call it the x-axis, you have the chemical reaction coordinate, and in the y-axis, you have the position or mechanical coordinate. And then you see that there is a potential energy surface that is displayed here, shown here in brown, which is defined by the interactions of the motor with its track and its fuel. A motor is essentially a device that is moving or diffusing on this potential energy surface. And notice that while in this part of the surface, it may be just doing chemistry because it's moving along the y-axis, there is a point where it actually has to move along in such a way that is in this slanted region where it has to do some movement at the same time that it does some chemistry. A motor essentially is a device that couples this chemical coordinate with this positional or mechanical coordinate. Okay, now What do we want to know about molecular motors? If we are going to study them, what do we want to know? We are interested in a number of properties. First of all, we want to know, for example, the type of operation of the motor. Is it a power stroke or is it a Brownian ratchet? And I will clarify those terms in a minute. What is the step size of the motor? In other words, what is its displacement for every molecule of fuel that it consumes? What is the maximum force or torque? Well, it's called often the stall force or the stall torque that the machine can actually generate. And that's important because allows us to determine what is called the thermodynamic efficiency of the engine. What is the mechanochemical cycle of the motor? In other words, what are the steps of chemistry and mechanics that actually constitute the cycle of the motor? How does its velocity relates to force? In other words, how does the velocity of the motor is affected? How much is it affected by load or force applied to the motor? This is important because it allows us to identify what is the step in which the actual coupling between chemistry and mechanics, generation of force, takes place. And finally, what are the parts of the motor and how are they regulated and coordinated during the motor's cycle? Okay, so for example, when we talk about power stroke versus Brownian ratchet, I like to start describing what a Brownian ratchet is. A Brownian ratchet, notice that I am here now using the mechanochemical space because one coordinate is a chemical coordinate and the other coordinate here is a mechanical coordinate. So in a Brownian ratchet, a term that was introduced by George Oster here at Berkeley several years ago, the motor essentially moves back and forth, oscillates, fluctuates along the mechanical coordinate, as I show here, without doing any chemistry. And then suddenly, when the motor is at that position, a chemical event happens that actually allows it to move along the chemical coordinate, but induces a change in the motor that prevents the motor from being able to go back. So now the motor is here. It has now moved by this amount along the mechanical coordinate, and it cannot go back. And now here it starts again fluctuating back and forth until a chemical event prevents it from going back, and then the motor has now made a progress from here to here along the mechanical coordinate. In a Brownian ratchet, essentially the chemical event rectifies the fluctuations of the motor along the mechanical coordinate. That's the Brownian ratchet. And RNA polymerase, which is the enzyme responsible for copying DNA into mRNA, is one example of a Brownian ratchet motor. The power stroke, on the other hand, is a motor in which at some point during the cycle of the motor does chemistry, but at the same time does mechanical coordinate progress. In other words, the motor is moving along this diagonal. So the motor cannot do chemistry without moving, and the motor cannot move without doing chemistry. That is the power stroke motor, which is also found in many motors in the cell. One example is ATP synthase in the mitochondria, where the motor, when it binds ATP, has a first power stroke. So that's the first chemistry going with power stroke together with power stroke movement. And then the hydrolysis, which is another chemical event, hydrolysis of ATP allows it to complete its power stroke, first of 80 degrees and then 80 plus 40, 120 degrees. Okay, so this is an example of the power stroke. Now, how do we go about studying these machines? I like to propose that the best way to look at these machines is to actually use force spectroscopy methods, such as optical tweezers, atomic force microscopy, magnetic tweezers, because the variables that are more easily detected by these methods, which are force, torque, displacement, and time, are also the ones that have the greatest functional value to understand the operation of these motors. Now, in my laboratory, we use optical tweezers. These are instruments that we built in my laboratory, and that actually we have worked very hard in order to obtain the highest possible resolution, spatial resolution, with this instrument to be able to study tiny little movement and displacement by these motors. So using one of these optical tweezers machines, which the optical tweezers are nothing but a laser beam that is actually focused through an objective of a microscope, and then that focused beam is able to trap matter. And so the matter that we trap are tiny little beads, plastic beads, and these plastic beads can be attached to molecules. And by moving and trapping the beads, we can manipulate the beads, and therefore we can manipulate the molecules that are attached to them. One example is shown here, where you see that we have two beads, one atop a pipette and the other trapped in an optical tweezers. And you see that by moving the trap of the laser, we are able to actually study the elastic response of a molecule, a single molecule of DNA. This is an experiment that we carried out many, many years ago in 1992, 91, 92, and that really gave origin to this field of force spectroscopy. Expanding on these initial experiments is possible now to study molecular motors, because if you have the motor represented here by this little green guy attached to a linker, and that linker is attached to a bead that is held in an optical trap, indicated here by this orange region of the beam, when the motor moves, it will tend to pull the bead out of the center of the trap. As it pulls the bead out of the center of the trap, it will actually make the direction of the light actually change slightly. By detecting that direction of the light change, we can determine the displacement that the motor made, and also determine the force with which the motor made that displacement. In other words, the force applied by the motor in order to move on its track, which is indicated here by this bar. As I said, this area of research is called force spectroscopy, and is what I'm going to show you the rest of this presentation. I'm sorry because I have a cold, a bad cold, and so my voice is not usually this coarse, but hopefully I will be able to finish my talk without too many problems. Okay. So the motor I'm going to tell you about is this one called the packaging motor of bacteriophage Phe29. Bacteriophage Bacteriophages have a capsid, the protein capsid, and in many of these bacteriophages, the motor sits, attaches to the bottom of the capsid, and its function is to package the DNA of the bacteriophage inside the capsid, okay? In this particular case, the motor is made up of three coaxial rings. Three coaxial rings. Here you can see it turn around. And the first ring is so-called connector, which connects the rest of the motor to the bottom of the capsid. It's a dodecamer. Then there is a ring of 5 RNA molecules, as shown here in fuchsia or magenta. And then there is a ring of 5 ATPases, which are the actual cylinders of our motor. In other words, if we can think of our motor, of our engine, as a 5-cylinder engine, right? Because it has 5 ATPases. And we can then do an experiment like is shown here. We can put a single bacteriophage that is in the process of packaging its DNA, right? attach it to the surface of a bead held atop a micropipette, and then we can grab the other end of the DNA and attach it via a streptavidin biotin to another bead, which is held in an optical trap, which is indicated here by these tick marks. When I add ATP here to the system, the motor will start packaging the DNA. And I can do two types of experiments. In the first experiment, which we call at constant force, as the motor actually pulls the DNA and packages it inside the capsid of the motor, it will tend to actually pull the bead out of the trap. But we can actually also just move, and of course, it will experience higher force because it's actually working against the trapping force of the laser of the optical trap. But we can make the motor work at constant force. And so we can actually move the pipette towards the trap so that the force doesn't change. And of course, the movement of the pipette gives us information about the progress that the motor has made in packaging the DNA, right? So this is what I show you here, where the tether length, of course, is going to get shorter because the pipette is getting closer in order to keep the force constant. And so the tether length gets shorter in time, as you see here. And I show you here 4 different experiments of the same type. Because we are plotting here length or distance versus time, we can think of these curves as the molecular trajectories of the motor, right? That's what a trajectory is in physics, distance versus time. And the slope of these trajectories is nothing but the speed of the motor, namely the rate at which the motor is packaging the DNA into the capsid. And the initial slope, you can see here, is about 100 base pairs per second. So this motor is packaging the DNA at about 100 base pairs per second. Now, notice that as the motor packages the DNA, eventually the slope actually gets smaller and smaller. So something is actually slowing down the motor, even though we are keeping the tension on the DNA constant. Keep that in mind. Why? Because, as you will see, we can do a second experiment in which we don't move the pipette and we just add ATP. And so what is going to happen is that the motor is going to pull the bead out of the center of the trap, and it's going to experience more and more force until it's working against a force that is so high that the motor can no longer package the DNA. That's what we call the stall force of the motor, right? And if we plot a histogram of the stall force of this motor, we find that it's a very powerful motor with an average stall force, fuerza de pare would be in Spanish, stall force of 55 piconewtons. But you can see that some of these motors can actually package at forces even greater than 70 piconewtons. And the question is, why? Why do we have such a powerful motor? To give you a way of thinking about this, the myosin, which is the muscle in our bodies, can only generate a maximum force between 3 and 5 piconewtons of force. So this motor being able to generate 70 piconewtons of force is really a powerful, powerful motor, right? And so it turns out that the explanation is that we have estimated that at the end of packaging of the DNA inside the capsid, the motor is working against an internal pressure of DNA inside the capsid of about 30 atmospheres of DNA, namely, or the same thing as 3 megapascals. Now, the reason is that the DNA is packed so tightly that the concentration of DNA at the end of packaging is about 400 milligrams per milliliter, which is essentially the concentration at which you find DNA when you crystallize DNA, when you make crystals of DNA. So obviously you need a very powerful motor in order to be able to package the DNA inside against 30 atmospheres of pressure. But of course, you will say, that doesn't make any sense because all nature had to do was to make the capsid larger and the pressure would not be so high at the end, and you didn't have to actually evolve a motor so powerful. But nature has another problem, and is that once the packaging is complete, the motor is taken out and the tail of the bacteriophage is put in. And now, after all the bacteriophages have packaged the DNA and are assembled, the Bacillus subtilis, which is the host bacterium, breaks and releases between 80 and 100 of these new bacteriophages that now go to infect other cells of Bacillus subtilis. As you see here, they sit on the surface of the cell, and now they have the problem of how do they actually put their DNA into the new bacteria? Well, there is no ATP in the outside, in the extracellular medium. They don't even have the original motor because it was taken out in order to put the tail with which they hope to inject the DNA. And so the solution is to actually use the pressure that has been accumulated in the head of the bacteriophage, 30 atmospheres of pressure, and then use that pressure to inject the DNA from the capsid into the host. Just for reference, I will mention that when you open a bottle of champagne, the cork comes out, I should say maybe cava or spumante, either way. Internationally, that wine has to have a pressure of carbon dioxide of 5 atmospheres in order to be sold as a bubbly. And so when you open a bottle of this sparkling wine, the cork comes out at the pressure of 5 atmospheres. The DNA inside the capsid of a bacteriophage is at 6 times that pressure. So that gives you a sense of how nature is very mechanical. It really has converted the energy of chemical energy of ATP, in this case, into potential mechanical energy in the force of a loaded spring of DNA. And then when the time comes, it can use that potential energy and convert it into kinetic energy and inject the DNA inside the cell. Okay, so we can now calculate the efficiency of this motor. We know that it can generate 70 piconewtons of force, and it can actually, we know how much is the step size, which I haven't mentioned here in great detail yet, but that allows us to calculate the work done by the motor. And since we know the energy of ATP in 1 ATP, then we can estimate that the efficiency of the motor at stall is about 63%. That's a huge thermodynamic efficiency. Our mechanical machines, the ones that we make that are human-made, can never be as efficient as these tiny little machines. And so that brings us to another question. Excuse me. Which is, what is the origin of the large thermodynamic efficiency of these tiny little molecular machines? Another way to ask this question is, from a physics point of view, is the following. If I have a system that is in state A, and the system is going to be moving from state A to a new state, state B, out of a non-equilibrium process, like all these machines are working in non-equilibrium conditions, then I can ask, what controls the amount of dissipation of energy dissipated in this process of going from A to B? Of course, if I want the process to be very efficient, I want to minimize the amount of dissipation. If I don't care about how efficient this process is, then I can just dissipate any amount of energy in the form of heat as I go from A to B. So in trying to understand what is the origin of this thermodynamic efficiency of these machines, I want to pose it in the way of answering this question. What controls the dissipation in going from A to B? Well, not too long ago, a theory was formulated by Sivak and Crookes that actually says that the excess work relative to equilibrium, the excess power dissipated in a given process actually should be proportional to a quantity, which is this zeta, which is a what they call a generalized friction coefficient multiplied by the speed at which we go from A to B, which is given by this control parameter lambda square, lambda dot square. So this is the derivative of lambda with time square, right? So in other words, the velocity with which we change lambda, but square velocity. And they also show that this generalized friction coefficient, which varies depending on where you are in the trajectory, namely with the control parameter, with lambda, depends or can be written and can be obtained from the equilibrium. Notice this is a non-equilibrium process, but can be obtained from the autocorrelation of the forces, of the fluctuations of force at equilibrium, okay? And so, and then finally, they then ask, what is the protocol that will allow us in going from A to B to actually minimize the dissipation in that trajectory? And they found, or they show that the trajectory that minimizes the dissipation is one in which the rate of change of lambda, in other words, the parameter, the control parameter, the velocity with which the system moves from A to B by its path, lambda, varies exactly proportional to the instantaneous value at that lambda of the generalized friction coefficient, but to the 1/2 power, in other words, to the square root of the value of the generalized friction coefficient. That is what the theory proposes. Okay, so of course, we want to actually study whether these trajectories actually show us that kind of change in velocity in order to actually optimize the efficiency of the motor. But studying these motors is not easy, as you know, and so we decided to actually test the theory first using a simpler toy model, and this toy model is a molecule of RNA, a hairpin of RNA, that is grabbed by its ends and attached to beads that are held in optical traps, as shown here. Two optical tweezers, each one holds one of these beads. And then the process is going to go from folded to unfolded, to take the system from folded to unfolded. And I can do that by pulling the beads and essentially opening the hairpin of RNA, right? Okay, so that is the idea. In this case, the control parameter is the distance between the beads, and the rate at which I separate those beads is the derivative of lambda with respect to time, in other words, lambda dot, right? Very good. So now I actually can do this process, and at the same time, I want to do the autocorrelation of the forces so that I can calculate the autocorrelation of forces, so I can calculate zeta, or the generalized friction coefficient for this system in going from the folded state to the unfolded state. And what I find is that at the beginning, the force is fluctuating, and I can calculate the autocorrelation of the force here. Then it starts fluctuating a lot. And then finally, when it is open, it fluctuates less. So by doing the autocorrelation, you can see that what I observe is that at the beginning, the generalized friction coefficient is small, then it goes up a lot, and then it actually comes down again in going from here to here. So if I'm going to minimize dissipation according to the theory, I should actually not move at a constant rate, but I should be at the beginning moving fast because the friction coefficient is small. Then when the friction coefficient increases, I should actually decrease the rate at which I move, my velocity. And then when it goes back to be small, again, I can come back to a higher velocity. And so this would be a clever protocol that should minimize dissipation, right, in this process, compared to a naive protocol in which I don't care. I just put at the average velocity. And so the idea is to compare the smart protocol with the naive protocol. And what you see here is that when you plot force versus extension, you see that the force increases until suddenly the hairpin opens. Then you continue pulling, then you relax the hairpin, and you see that the hairpin falls back at the lower force, and so there is hysteresis. And this hysteresis, the area between these two curves in red here, is in fact the amount of energy that has been dissipated in the process. And notice that when I use the naive cycle, I have a lot more dissipation than when I use the optimal or the minimum dissipation cycle protocol that I just described. And in fact, you can see that the region where maximum dissipation occurs is in the middle, and you see that the naive protocol dissipates a lot of energy there, whereas the clever protocol actually is doing much less dissipation. And so I may generate a conjecture. My conjecture is this: have molecular machines of the ones we've been talking about evolve to harness energy into useful work, minimizing dissipation of heat. In other words, in the case of the bacteriophage that I have been showing you, when the motor starts actually filling the head, the capsid of the bacteriophage with a lot of DNA, we begin to see very interesting behavior. The motor starts generating long pauses, or it starts moving very slowly before it actually, and then there are regions in which it moves fast again, and then slow and fast again, and you can see this in all these different traces, making us wonder whether in fact this is a manifestation of the ability of this motor to actually control the amount of dissipation by adapting the load, adapting the rate at which they actually carry the work proportional to the friction or load that they experience. Another example is a motor that we are studying in the laboratory too, which is the ribosome. The ribosome, as you know, is responsible for synthesizing proteins in our cells. And we can actually do an experiment like this, where we actually follow the movement of a ribosome over the messenger RNA, and we can see how the ribosome invades a messenger RNA, and we can follow that invasion by how the distance between these beads increases, because whenever the hairpin opens, the distance between the beads will increase if we keep them at constant force, at constant tension. And what we find is that the motor, we can actually follow the ribosome moving at with the resolution of 1 codon at the time, that if the hairpin is a very I would say, easy to open hairpin, then the motor will actually be going 90% with a high speed, what we call the fast pathway, okay? And it will go by this path. And then only 10% it will go through what is called, we observe that is a slow path. So the motor has the ability to actually have 2 different gears, a fast gear and a slow gear, and in front of a barrier that is not too difficult to open, has little GC content, the motor goes 90% this way, 10% this way. But when the motor encounters stronger secondary structure, now what we see experimentally is that the motor goes 50% of the time by the fast pathway and also 50% of the time by the slow pathway. So the motor changes gears, so to speak, in order to actually face the harder barrier that represents a more stable RNA hairpin. Okay, now, so this again may be a manifestation of what I'm just telling you about and explain why these machines are so thermodynamically efficient. But let me go back to the bacteriophage Phe29 motor. We eventually developed a very high resolution instrument that allow us to actually do an experiment like this using a dual trap optical tweezers machine. Again, we have the capsid of the bacteriophage here is packaging its DNA. I grab the end of the DNA and attach it to another bead, which is in another trap. And then I add ATP. And this is what we see at very high resolution. Remember, we had a trajectory that was very continuous. Now we are going to see trajectory, but now we can see the details of that trajectory at high resolution. And you will see, oops, for some reason, this is not playing. Oh, well, let me try again. Sorry. Oh. All right. Let me try again here because it's okay. Okay, let's see. Now it's working. I think. Oh, it's kind of stopping for reasons I don't understand. But anyway, here is what is happening. And then you see that we are going to see the trajectory. There is the trajectory, right? The motor, and we are doing it at constant force. But the trajectory, we have enough resolution so we can see that the trajectory actually is made up of periods of dwells where nothing happens, and then bursts when the motor actually packages DNA, and then dwell again and burst, and then dwell again and burst, and then dwell again and burst, right? Okay, so that is what we observe in this experiment. And in fact, I can show you here in this slide that these trajectories, which are made out of dwells and bursts, dwells and bursts, dwells and bursts, right? Dwells and bursts, dwells and bursts. Now, what I have done here is I have changed the amount of ATP that I give to the motor. In the black curve, the motor is working with 5 micromolar of ATP, and I start increasing the concentration of ATP as I show you the other trajectories, okay? Notice that the burst, which is 10 base pairs because it has the distance between these dashed lines, the distance between the dashed lines is 10 base pairs, is independent of the concentration of ATP. At very high concentration of ATP, I still have bursts of 10 base pairs. Just as I see at 5 micromolar, at very low concentration of ATP. What changes is that the length of the dwell decreases as I increase the concentration of ATP, right? Indicating that the binding of ATP to the motor occurs during the dwell. When there is little ATP in the medium, the dwells are long because the motor takes a while to actually saturate itself with ATP. When there is a lot of ATP in the medium, the dwell is very short because ATP binds quickly to the 5 spots, the 5 subunits in the motor, and the motor is ready now to translocate by 10 base pairs, okay? So if I told you that this is a pentameric motor, a homopentameric motor, all 5 subunits are identical, and I tell you that the step, the burst, is 10 base pairs, and I ask you how many base pairs are packaged by each one of these subunits or ATPases, naturally you will say 10 divided by 2, 10 divided by 5 is 2, and therefore each subunit should be actually packaging 2 base pairs, right? Okay, that's what we expected. And then my student one day came and said, look, what we find is that the steps, the sub-steps, each one of these 10 base pairs is made up of 4 sub-steps of 2.5 base pairs. Of course, I was very worried by this and troubled by this result because I knew that there were 5 ATPases and they were telling me that only 4 of the ATPases were working and not doing 2 base pairs, but 2.5 base pairs. Okay, so I asked my student to go back and I said, we are never going to be able to publish this. So you better repeat the experiment very carefully. He left, he came back 3 weeks later and said, you were right, it is not 2.5 base pairs, it's 2.4 base pairs plus minus 0.1 base pair, which of course established that the motor indeed packages in 4 steps of about 2.4, 2.5 base pairs. That means immediately that one of the ATPases is not doing any work. 4 of the ATPases, which are all identical, by the way, 4 of the ATPases are doing work, but one of them is not doing any work. My students immediately called the 5th ATPase the PI, right? Obviously. And so, but it became established that actually there was some way of symmetry breaking in the pentameric motor that made one of the ATPases behave differently. So I can show you here the mechanochemical cycle that we have here. The motor is in a dual, binds ATP, and then makes 4 steps of 2.5 base pairs during the burst. And then it goes into a dual again where it exchanges ATP for ATP and is ready to actually do the next burst. So one of the subunits is distinct and does not perform a mechanical task, but probably a regulatory task. Well, I'm going to make a long story short and show you what we have learned about the motor since then. Actually, we know that it's a very powerful motor. I told you already, can generate forces of 70 piconewtons. We know that the power stroke coincides with the release of phosphate during ATP hydrolysis. The ATPase activities of the subunits are highly coordinated. I think it's clear. I showed you that the packaging occurs in cycles of duels and bursts, right? And each burst is 10 base pairs made up of 4 2.5 base pairs steps. The motor, it turns out, we showed, makes critical electrostatic contacts with the DNA that is being packaged every 10 phosphates. And these electrostatic contacts have both load-bearing as well as a regulatory function for the motor. And one subunit is special. It doesn't carry out a mechanical task. So here is a rendition of how the motor actually does the work during the duel. 1, 2, 3, 4, and 5 ATPs bind. And then the special subunit is the first one to hydrolyze ATP, but it doesn't translocate. And then orders the other subunits to hydrolyze and translocate the DNA in steps of 2.5, 2.5, 2.5, and 2.5 base pairs before the motor enters a new duel. And so again, here, even though only 4 subunits translocate the ATP, the 5th subunit we actually show, I don't have the time to show it here to you, also bind ATP and hydrolyzes ATP. And here,
Carlos,
yes.
Sorry to interrupt. So just I'm noticing the time, and it would be great if you could have time for questions. So I was just wondering if
yeah, I normally give this talk much faster, but I'm worrying about not being able, people not being able to translate. So I'll speed up, and I need 2 more minutes.
Perfect.
Okay, so here is the full mechanochemical cycle of the motor. Notice during the duel, there is exchange of ATP with ADP, as shown here. Then the special subunit hydrolyzes, and tells the other subunits start hydrolyzing and translocating 4 steps of 2.5, and then a new dwell. And the question now is, what breaks the symmetry of the ring? And so what makes the special subunit special if they are all identical? We found the answer asking a completely different question, which is, does the motor rotate the DNA while it translocates it? And in order to answer that question, we actually designed this experiment that you see here, where we have a bead on the pipette and a bead on an optical trap, but now we put a tiny little bead on the side of the DNA. And we ask, does the motor actually rotate the DNA? Does it generate torque? And so here is what we observed when we actually added ATP. Of course, the motor, this is not working very well, but there it is. You can actually see that as the process occurs, as the packaging occurs, the motor is actually rotating the DNA, and that is seen as a rotation of the tiny little bit. And so the answer to the question is yes, the motor can generate torque and can generate force, as you see, 70 piconewtons. And when we asked, does the motor rotate the DNA by how much, we found out that it was 1.5 degrees per base pair. And it turns out that 1.5 degrees means the following. The motor makes contacts every 10 base pairs with the DNA, and the special subunit probably is the one that actually contacts the phosphate in the DNA. After 10 base pairs of packaging, the special subunit is looking for its phosphate, but it cannot find it. And so it has to rotate the DNA exactly by 15 degrees every 10 base pairs, or 1.5 degrees per base pair, in order to contact again the phosphate and retain its character as the special subunit. And that is why the motor has to rotate the DNA in order for the special subunit to retain its identity as the coordinator of the whole cycle of the motor. So let me just show you a movie, which probably is not gonna play again here, although maybe it will. Yeah, it is. I don't know why it is actually stuck this way, but in any case, there it is, the motor. There is the motor. You can see at the end of the trap, during the dwell, there is exchange between ATP and ADP. This subunit binds ATP, the other releases ADP, binds ATP, releases ADP, binds ATP, releases ADP. And finally, the special subunit in green is the one that first hydrolyzes and then doesn't do anything, but just tells the other subunits, okay, start hydrolyzing and translocate it. 2.5, 2.5, 2.5, 2.5, 2.5. That way, where is my phosphate? The special subunit rotates the DNA by 14 degrees, and then the cycle starts again. Okay, that's the end of my presentation, and I will now give you just the acknowledgements, well, maybe now it's running, so maybe it's worth just seeing it running. Where is there is a 14 degrees rotation, you'll see here at the end of this. And okay, well, anyway. So let me just say this is the people that did the work over the years, and I will just stop here because of time. Thank you very much.
Thank you, Carlos. That was an absolutely fantastic talk. I don't know if there are questions from the audience right now. We have time maybe for a couple of questions. And as I mentioned at the beginning, it can be both scientific or about Carlos's trajectory if you're interested. I have a quick question while people are getting the courage. So how difficult is it to establish? I mean, how did you get interested in this area of science, and how difficult has it been to establish everything that you have been using for this type of work?
Yeah, thank you for the question. Well, look, what happened to me was that actually I originally trained as a theorist here at Berkeley, and then at the end of my doctorate, I actually, my professor offered me to do a postdoc while I was looking for a job. And because I tried to go back to Peru, and unfortunately at that time, the country had just started the war. That was really a civil war with Shining Path, a Maoist guerrilla group. And the last thing they needed at that moment were biophysicists like me. So there were no job for me. And so I came back to the United States, and my professor offered me to do a postdoc. I did a postdoc, but it was experimental postdoc. And then I had to look for a job here, and the only job that I got was at the University of New Mexico in Albuquerque. And being there actually helped me a lot because I was really very isolated in a sense, and I could think of the crazy experiments that nobody had done. And one of those crazy experiments was to actually grab a single molecule of DNA by the end and actually pull on it, just so that actually we could learn about the elasticity of a molecule. It was a fun experiment. And that was the beginning of this force spectroscopy field. And we discovered that the best way to go about it was to build our own instruments and perfect them more and more every time, so that actually we could make experiments that had not unedited experiments that had not been done ever before. And that's how we got into this field. Yes.
That's very interesting. I mean, I think it's inspiring also for younger people to see how one gets there, right? To see how it sounds. I mean, of course, you are a super well-established scientist, and then for someone starting, it can be like, how do you get there? But that's a great response. I don't know if there are others that want to ask any questions. Otherwise, do you agree that, Carlos, if we share your contact details with, participants here and others that might not have been able to attend, so they can get directly in touch with you with more questions?
Sure, I'll be happy to actually answer questions and share my contact. I am very interested in any way possibly help students from Spanish-speaking America and as well as Brazil to actually who are interested in science. I think that this is a wonderful job, and that it is possible to actually make a really important contribution to our understanding of nature by actually doing science. And we Spanish-speaking scientists should actually have to be convinced that we can do it, si se puede. Si se
Absolutely.
puede, and si se puede here in the United States, and si se puede back in the Patria Grande, in South America, in Central America, in Mexico. It can be done. It can be done, but it requires a lot of dedication, a lot of effort, a lot of discipline, personal discipline. That's a very important thing, to have discipline, have well-defined objectives in your career, in your scientific, in the design of your scientific career. And in that way, you can actually make progress and in some cases succeed.
That's a very good message. And this talk will have been recorded, so I will share the link with everyone who couldn't attend today. Yes. With this, I think I'd like to thank you, Carlos. You're amazing. I'm a big admirer, and I thank you for participating in the mini series.
My pleasure. My pleasure. Thank you very much for inviting me.